Hey there, welcome to Dr. Clausen's dystopian. He tries to lecture while everybody tries to get something from him, including a cat apparently. um, so here we have this is where we left off. We have the malate dehydrogenase catalyzing the oxidation of malate. This yields oxaloacetate and the NADH. And this is an equilibrium right here. So remember, that's an equilibrium step. The delta G here, the delta G naught prime actually is significantly positive. This is 29.7 kilojoules per mole. And so what you're seeing here is that this would not normally go forward spontaneously. We would consider this very, very stacked towards the left side, the reactants. But remember that delta G naughts are if everything's essentially at their normal conditions. If you continuously take away one of the products, then you're going to pull an equilibrium towards that direction. The big thing is that you have to be able to establish the equilibrium, and you have to have a fast enough reaction going back and forth that it actually happens within some sort of period of lifetime that makes sense. It happening in 100 years obviously would be ridiculous. So again, this is actually going to be driven by the use of the products. So the oxaloacetate is actually going to be used by the citric acid citrate synthase specifically. And then the NADH is actually going to be used up in the electron transport chain. And this is actually basically, the NADH part is actually usually linked. It's happening pop, pop, pop immediately. That's very fast. The net reaction of your citrus acid cycle would actually be this reaction right here. So this is acetyl-CoA. I don't know if you guys can hear that, but my cat is just going crazy and playing with a toy in the background, making a hell of a lot of noise. So the acetyl-CoA, this plus 3 NAD plus FAD, notice those again are the electron transporters. Your ADP plus a orthophosphate, that'll give you the ATP, and 2 waters. Now, an interesting side note to this is that, directly speaking, this citric acid cycle produces very little ATP. Though, considering that you're only using a single acetyl-CoA to do this, that's not that bad. But, yeah, this is not actually directly, without the electron transport chain, producing a huge amount of ATP. So 2 CO2s are produced in the products. You have 3 NADHs, FADH2 of course, and then ATP and 2 H pluses and a CoA. And it's very important that it's recycled because these are actually quite expensive. You'll notice they are actually relatively complex and large, and they take quite a bit of energy to make actually. So your 2 carbon atoms that enter each cycle from the acetyl-CoA right here are not actually the ones that leave as CO2. Typically what happens is this is from the previous cycle that are chopped off. And I think I had mentioned that actually in the actual citrus acid cycle. Sometimes get ahead of myself. So during the initial 2 decarboxylation reactions, that's when you actually chop off those carbons. So 2 carbon atoms are going to enter the form of acetyl-CoA, and 2 carbon atoms are going to leave in the form of CO2 molecules. Again, keep in mind that you are not actually, you actually end with the carbons from this, in this products here. You don't actually lose the, like these carbons are not these ones right here. So those are from the previous cycle. Those are the ones that got fed in at the beginning. So, again, you have 2 carbon atoms in, you have 2 carbon atoms out, because you have to have the same starting conditions over and over. This is why, by the way, that it didn't mention the beginning of the citrus acid cycle, that second substrate, because it would show up on both sides of this reaction, and so it cancels out. Though that is not actually the same substrate, it is not the same atoms, well, not the same carbon atoms at the very least. I believe there is a possibility of a few of the oxygens being maintained, actually. The 4 pairs of hydrogen atoms, leave in the 4 oxidation reactions. This will yield your 3 NADHs and 1 FADH2. If you remember from that cycle where we went after it, which is probably the most important part of the chapter, the FADH2 reaction actually happens because you don't have enough energy for the NADH. So that actually is a meaningful difference there. Most people tend to ignore that and just be like, it could have been NADH or whatever. But these are meaningfully different. It's like having a big industrial capacitor versus a small little capacitor you'd have in like your car or your house or a computer or something. You would never be able to actually use the other one. It wouldn't do you any good, or at least it would destroy your house if you actually did use it. So one compound with high phosphoryl transfer potential, usually ATP, is generated. 2 water molecules are going to be consumed. When you oxidized versus via the electron transport chain, each pair of electrons, now we will actually cover the electron transport chain, but so you understand the overall results of what we're looking at, each pair of electrons from the NADH is going to generate 2.5 ATP on average. Each pair of electrons for the FADH2 is going to generate approximately 1.5 ATP, again on average. This is approximation numbers. Keep in mind that different organisms, even down to individuals, can be much more efficient at this process. And so you could have an organism that generates 2.6 ATP here. It could generate 1.4 ATP here, though. Or it could be 1.6. Or it could, I believe the differences that have been found have been up to like a, it wasn't 20%, I don't think. It was something a little bit lower than 20%. It's been a while since I saw the paper. I can try to look that up if anybody's actually interested. So you have 9 high transfer potential phosphoryl groups. These are generated from the oxidation of the 3 NADH and 1 FADH2 molecules. 1 ATP is going to be directly formed in one round of your citric acid cycle. Or keep in mind that you do have access to the enzymes to interchange these ATP, GTP, etc. And so you have one equivalent that's formed that you have actually available. There is a physical association of the citric acid cycle enzymes into the supramolecular complex. This actually allows for substrate channeling. And so this is the overall cycle and the reactions that make that up and the enzymes for each step. This is actually how I probably would recommend trying to memorize this. You will need to memorize this. This is a central tenement of all biochemistry, and by biochemistry I mean literally if you're going to do anything with any one or any organism on a molecular level, meaning give them drugs, you really should know this. This is like psychological disorders have actually been linked to the mitochondria and its different problems with the mitochondria. There's a very strong evidence that at least 30% of psychological disorders are just straight up mitochondrial diseases, and probably more, but those are just the ones they could actually identify because we actually don't know that much about how to regulate the mitochondria and drugs to do that and, try to fix things. And this is really new research, but it is pretty solid with 3 different groups all finding the same results in different ways. So it's pretty wild. The other one being the gut bacteria is kind of one of the other new ones that hasn't really been well understood yet. But yeah, it's surprising how little of what's going on is actually the DNA and your normal mechanisms that we've discovered so far. It's quite complex, actually. As you probably surmised from my metabolism web I keep talking about. Right here, this is again your citric acid cycle put in a tabular format. So right here you have your different steps. And then you have your reactions, where it breaks up the reactions, and that's important. That it has a 2A, 2B here. This is the enzyme that goes along with that. Keep in mind that's usually the substrate and what it does, though every now and then you do get things where it's the enzyme for the reverse on these cycles and these pathways. So this is then the prosthetic group that goes along with it. This is actually some of the most interesting stuff with this prosthetic group, because it's something that's only recently been able to be understood, because the work you have to do to figure out the prosthetic groups for each of these enzymes and what they look like and all that is pretty wild, actually. This is the reaction types. Now, this is one thing that you can definitely tell this is a biochemist doing it, because there actually is shortened forms of every single one of these reaction types in organic chemistry. And yet they used A, B, C, D, and then put them down here when they could have just put the shortened form. Yeah, whatever. Anyways, condensation A. B is dehydration. C would be hydration. D is decarboxylation. E is oxidation. And F is substrate level phosphorylation. Keep in mind that the oxidations can go along with other things. For example, the decarboxylation is going to have oxidation with it. This right here is the delta G, not primes. I do find it amusing that they always put these together. You really could have just put this with this, and that's how it's normally done in most publications, except for biochemistry and biology, where they do them separate, which is kind of amusing. All right, so the entry to the citric acid cycle in your metabolism, this is going to be thoroughly controlled, because once you commit to the citric acid cycle, it can be very hard to go backwards, specifically because you have times where you're going to lose CO2, and even before you commit to the citric acid cycle. And so this losing CO2 portions, I can't say you can't go backwards, because theoretically you could, but the likelihood of you having a high enough concentration in your cell of CO2 to ever go backwards is, to go backwards any significant amount, I should say, is very, very, very low. It's probably never going to go backwards. So the formation of acetyl-CoA from pyruvate is irreversible in animal cells. The acetyl-CoA has two principal fates to it: your metabolism by your citric acid cycle, and incorporation into various different lipids, specifically a lot of times fatty acids. Remember that the fatty acids actually came in at the acetyl-CoAs, not up here at the pyruvates. The activity for the pyruvate dehydrogenase complex is going to be tightly controlled allosterically. This is also by reversible phosphorylation. And so this is typically the point at which you control this, because after this step it becomes very, very, very difficult, if not soft cap, kind of impossible, under physiological conditions, to go backwards to your glucose. And remember, glucose is one of those key molecules that's important for making more complex molecules, other sugars, which we'll get into in the 5pp, and feeding your brain, heart, red blood cells, etc., giving them energy. So anyways, that's the acetyl-CoA, by the way. Going off to CO2 is typically the citric acid cycle, and then fatty acids would be your fatty acid lipids. Highly concentrations of reaction products are going to inhibit the reaction by informing the enzyme that there's no need to metabolize pyruvate to the acetyl-CoA. The acetyl-CoA will inhibit the trans-acetylase component, which is your E2. Your NADH is going to inhibit the dihydrolipyl dehydrogenase, which is your E3. And remember, those are those different domains we had talked about before. So the pyruvate dehydrogenase kinase, the PDK, is going to phosphorylate the pyruvate dehydrogenase component, which is E1. This is going to switch off the activity of the complex. Pyruvate dehydrogenase phosphatase, PDP, will dephosphorylate the E1. So in mammals, both the PDK and PDP are going to actually be associated with the E2, E3, BP core complex. And so they're actually going to be associated and typically incorporated with those complexes. All right. So this right here is the activity of your pyruvate dehydrogenase array being regulated by that reversible phosphorylation. And again, here we have a kinase coming along. It's going to, that's that kinase. Let's see, we're on the pyruvate dehydrogenase right here, PDK. So that's the kinase PDK coming along. We're using up ATP to ADP to give our phosphate group, which is then going to phosphorylate the PDH complex. That's going to give us the inactive complex. The phosphatase is the one that, remember, phosphatases are different than kinases because kinases transfer phosphate from ATP or onto ADP. And phosphatases are going to take the phosphate and cleave it using a water. So this is actually going to give you the active PDH, the dehydrogenase, and that's when you cleave the phosphate off. You dephosphorylate it. So this is actually going to also, the pyruvate dehydrogenase complex is going to respond to the changes in energy charge of your cell. This is actually that feedback activation and inhibition, activation here. So this is again that pyruvate to acetyl-CoA. This is the PDA, which is the pyruvate dehydrogenase right here, enzyme right here, the complex being acted on. And normally it go NAD to NADH. What's going to happen though is that you'll see that the NADH concentration, as that increases, it's actually going to inactivate this and inhibit its activity. The acetyl-CoA is going to inhibit the activity of your PDH as well. And this ATP here, which is actually, again, citric acid cycle coming down, is going to eventually, and we haven't gotten to it in great detail yet, but we've talked about electron transport chain and produce a great deal of ATP. And so when we make more ATP, that's also going to come back and give feedback inhibition on your PDH. What you can see here is during the low energy charge or very high pyruvate concentrations, those are actually going to activate the PDH. And ADP will also act to increase the activity of the PDH. So at rest, your ratios of NADH and NAD+, the acetyl-CoA versus the CoA, and the ATP versus ADP are going to be high. This is going to promote your phosphorylation and inactivation of the complex by activating PDK. And again, that's the methodology of this. Okay. During activity, you are going to increase your ADP, and so you'll have high ADP. Your pyruvate is going to activate the complex by inhibiting the kinase. The calcium 2+ is also going to stimulate the phosphatase, which will enhance the pyruvate dehydrogenase activity. And so, yes, I know that the arrows are going to PDH. This is to better inform you about the activity on the pathway. But the mechanism of this change in our activity is actually through this cycle here, which is the inactive versus the active forms of PDH through phosphorylation. And so remember, if you lower the activity of the kinase, that's going to cause less of this to be made from this, increasing the concentration of your active form. If you lower the activity of phosphatase, you get the reverse. The kinase will still be, taking the active PDH and making it inactive through phosphorylation, while the phosphatase will not be cleaving those phosphates off as fast, so you'll build up this form. Okay. I think you guys got that. In the liver tissue, the epinephrine is going to bind to the alpha-adrenogenic receptor. This is going to cause an increase in the calcium concentration. This will activate the phosphatase. And so we have actually now connected back our 7-TM all the way through to our citric acid cycle and all the way through into our mitochondria. So in the liver, the adipose tissue, insulin is going to stimulate the phosphatase. Remember, phosphatase is the bottom. Right there. Okay. So individuals with pyruvate dehydrogenase phosphatase deficiency have a pyruvate dehydrogenase complex that's always phosphorylated, i.e. the inactive form. In those individuals, glucose is going to be processed to lactate rather than to acetyl-CoA. This is going to result in a few things, but resulting in lactic acidosis, because we're going to have a buildup of lactate, of course. Many tissues are going to malfunction in the acidic environment. This will include the central nervous system, which is going to be inhibited by that acidic environment. It's going to have have a lot of things wrong with it, but it'll have a reduced function in the acidic environment, but it'll also have a charge differential, which it should not have. Diabetic neuropathy, this is going to be numbness, tingling, or pain in the hands, arms, fingers, toes, feet, and legs. Some of the common application, about 50% of patients of both type 1 and type 2 diabetes will eventually exhibit diabetic neuropathy. This can be treated with painkillers but cannot be cured. Overproduction of lactic acids by cells in the dorsal root ganglion may actually be a significant contributor to this. Typically, this happens due to uncontrolled diabetes, though it's hard to be controlled all throughout the day, all day, every day, if you do not have a functional insulin pathway. So hyperglycemia, this is high glucose concentration. This is the defining feature of diabetes. It increases the PDK activity in the cells of the dorsal root ganglion. It's going to lead to inhibition of the pyruvate dehydrogenase complex. The inhibition of the complex leads to the pyruvate being processed to lactate. Excess lactate is going to lead to an increase in the acid-sensing nociceptors, which are the which are the pain receptors. Excuse me. This is a type of G protein coupled receptor. This can result in diabetic neuropathy. So the isocitrate dehydrogenase and the alpha-ketoglutarate dehydrogenase are going to be allosteric enzymes that are going to primarily regulate the rate of cycling. These are the first two enzymes that harvest high energy electrons in your cycle. So again, isocitrate right here, isocitrate dehydrogenase, alpha-ketoglutarate, alpha-ketoglutarate dehydrogenase. And again, those are the allosteric enzymes that are primarily regulating the rate of cycling. Remember that the previous one we did is not actually an allosteric site regulation, it's actually a phosphorylation that's happening. So the citric acid cycle is going to integrate many of the cell's other metabolic pathways. It's going to include those of, again, it's the same pattern you may have already kind of figured out. But overall, what's going to happen is we're going to increase cycling as we have higher concentration of ADP. We'll decrease We will decrease the cycling as we have high concentrations of ATP or NADH. Remember, the NADH, if it's building up, means that we can't proceed along the electron transport chain, and so we got to slow this down to let that catch up. And ATP means we have too much energy, we don't need it anymore. We can leave other stuff. We can leave the adenosines for other stuff. Right here we have the alpha-ketoglutarate. This is actually kind of an interesting one because you get a slight difference in activation and inhibitions. So right here we have ATP, again going to inhibit this. Remember, this is allosteric inhibition, so it's actually a perfect use of inhibition. You'll notice, by the way, that imperfect uses of inhibition are very common. The succinyl-CoA is also going to act to inhibit this, and again, that's actually feedback inhibition, because that's the product. And NADH is also going to have a feedback inhibition, and again, that's too much NADH, we need to let it catch up on the electron transport chain, or it could indicate we have enough energy. Don't need to go forward. The succinyl-CoA, if I had to, an interesting little theory I have is that this is actually more to do with the CoA than it is the succinyl-CoA, the actual product. That the CoA is necessary to continue doing things like glycolysis and feeding this acetyl-CoA in here. And you need to recycle these and use these again to do stuff. So you can actually end up with a situation where you'd be low on CoA, not be able to go to the succinyl-CoA and continue the pathway. You don't want to use up all your CoA doing and build up a bunch of succinyl-CoA to sit there. Also, the thioester bond is quite high energy, so that would be a big waste of energy because that would hydrolyze if it was left sitting there. The citric acid cycle, this can integrate many of the cell's other metabolic pathways, including those of the carbohydrates, fats, amino acids, porphyrins. This tends to be sort of the central cycle everything kind of gets fed into. Many citric acid cycle components are going to be precursors for biosynthesis of key biomolecules. Again, here's your citric acid cycle, but how it's linked up to everything else. So again, this is that central important role in biosynthesis. This is why I was saying that. Of the things I've taught you, this is one of the most important things for you to actually be able to understand, because this is how everything else interplays. Like, for example, if you understand this pathway, you can actually understand better why things like if you want to treat seizures, you can actually have somebody fast or go on to a ketogenic diet, and they have a very high chance, one of the highest treatment methods of actually treating their seizures long term. I believe it was 30 or 40% region. You can also do things like inactivate some by going on to a ketogenic diet, very strict ketogenic diet for 3 to 4 months, I believe it was. You actually have a pretty good chance of treating, pretty good in the fact that autoimmune diseases usually are pretty tough to treat, at least long term. Fasting or ketogenic diets actually have a pretty good chance. It's a little bit under 20% if I remember correctly. Last time they did a study of permanently treating an autoimmune disease. Anyways, so this right here is how all of these things interact. Right here we have pyruvate, of course, going to acetyl-CoA, going to citrate. That's the normal cycle. But the citrate can actually go off to fatty acids. They can also make sterols. The alpha-ketoglutarate, this can make glutamate, other amino acids from that, and purines. It's like the DNA, RNA purines. This is the porphyrins right here, and the heme, and the chlorophyll. These are all derived from succinyl-CoA there. Oxaloacetate is going to go to aspartate. Other amino acids from that can go to purines, and this is actually where you start getting pyrimidines as well. And this right here, oxaloacetate, can go to glucose. And this is why I was mentioning that you can make glucose from fatty acids. You do not need basically any sugar at all in your diet whatsoever, and no carbs. Because you can go into the acetyl-CoA here from the fatty acids, go through the cycle, then go to glucose. It does some interesting chemistry to your body, whether that is long-term good or bad. So far, it looks like it's actually a positive thing on the vast majority of people, but I'm not gonna come back in 10 years and tell Dr. Costin that's wrong, please. I actually would be interested to find that out, though I'll probably have already read the article. But still, come back and talk to me. We'll talk about it. Pyruvate carboxylase is right here. This actually can go directly to the oxaloacetate if you need it, so you can make more glucose, kind of shortcutting the pathway. Or, amino acids, purines, pyrimidines, all important, like if you needed to make more DNA or RNA. The citric acid cycle intermediates, these must be replenished if any are used for biosynthesis. This is one of the reasons why you had all of those checkpoints where it'll stop if we have too much stuff here, if we're low on energy, if we're low on this, because basically a large portion of this pathway actually produces useful stuff. You don't actually want to digest every single component of this if you don't have to. You're going to need a large number of those. I mean, you need a lot of hemes for all those red blood cells, and for several other purposes actually as well. Though I don't think I'm actually going to be able to get into all those. The mammals lack the enzymes. I know I'm not going to be able to get on all those. What am I saying? The mammals actually lack the enzymes for the net conversion of acetyl-CoA into oxaloacetate or other cyclic intermediate. Yeah, so we actually have to go all the way around, actually, if we are in the acetyl-CoA, which is kind of an interesting thing. Rather than taking a shortcut anywhere in this pathway. There are creatures that can do that. Anaplerotic reaction. This is a reaction that will lead to the net synthesis or replenishment of your pathway components. Pyruvate carboxylase is going to catalyze the formation of the carboxylation of the pyruvate to the oxaloacetate. This reaction is actually used in gluconeogenesis and is dependent on the presence of acetyl-CoA. So this is the overall reaction for that, by the way. This is the pyruvate going directly to the oxaloacetate. Man, this is a great time that I could have helped you guys gain a little bit of critical thinking skills. Okay, I'm going to say it, pause the video, try to think of your own, okay? Pyruvate plus CO2 plus ATP plus water gives you oxaloacetate, ADP, orthophosphate, and 2H pluses. Notice you consume energy. This is actually not a great thing to do because going into the pathway, typically you're going to make more energy. But why would an animal need this? Why would this be extremely important to any organism that does the citric acid cycle? Okay, pause. Think a little bit. Think. Okay, hopefully you came up with something along the lines of, well, what if you don't have any oxaloacetate? What if you used up all of this or you just locally don't have it? If you can't get any oxaloacetate by the time that pyruvate hits a pyruvate carboxylase and actually goes through the reaction, then it will generate its own oxaloacetate. You can thus start a new cycle with another pyruvate and the new oxaloacetate. Acetyl-CoA, technically. Going along the citric acid cycle, generate more energy, and you can keep going. If you did not have this reaction pathway, technically you could get in a situation where you used all of your oxaloacetate, you made glucose out of it, whatever, amino acids, doesn't matter. And you have pyruvate going to acetyl-CoA, and you literally have no way of kickstarting this. You're just fucked. You're done for. Goodbye, done, dead. Okay, so interesting little side note, and a good chance of sort of building up your ability to think through these things. What is the consequences of this? Beriberi, this is a neurological and cardiovascular disorder that's caused by a dietary deficiency of thiamine, which is the vitamin B1. Thiamine is the precursor of the cofactor thiamine pyrophosphate, TPP. TPP is the prosthetic group of these three. Pyruvate dehydrogenase, alpha-ketoglutarate dehydrogenase, and the trans-ketolase. This trans-ketolase we haven't actually hit yet. We'll hit that in the pentose phosphate pathway. This is an enzyme in that pathway to convert different sugars. So the patients with beriberi have a higher than normal levels of pyruvate and alpha-ketoglutarate in their blood. Both mercury and arsenite, which is this right here, ASO3 3 minus, have a high affinity for neighboring sulfhydryls. So binding of mercury or arsenate to the dihydrolipoyl groups of the E3 complex, the pyruvate dehydrogenase complex, actually inhibits the complex and leads to central nervous system pathologies. Treatment is the administration of sulfhydryl reagents with the adjacent sulfhydryl groups compete with dihydrolipoyl residues for binding. Again, remember how I said that nervous system conditions and neurological disorders are incredibly common, actually, and very heavily linked to these reactions within the mitochondria, specifically the citric acid cycle being one of the more important ones, though, again, the mitochondria are incredibly crucial. This mercury poisoning here, I'm not sure if you guys are aware of this, but mercury poisoning is actually, if you remember Alice in Wonderland, the Mad Hatter, The Mad Hatter actually has mercury poisoning. That's what makes him like he is in that novel, which was then made into a movie. And so that was literally Alice in Wonderland is actually a statement about culture of that time, specifically English culture. And the fashion of the day was those large, incredibly complex hats that required mercury to actually make those, to give those the proper stiffness, that they could be made in that fashion. And so the Hatters had to heavily use mercury. They got mercury poisoning, of course, caused things like shakes, sudden bouts of fits, and a variety of other things. I'm not going to go into all that, I guess. Anyways, this is the arsenite, also has a similar mechanism. So here's the arsenite, by the way. It's actually going to inhibit the pyruvate dehydrogenase complex. And so here's your dihydrolipoamide group right here. And that's that sulfhydryl, excuse me. That's again going to be on the pyruvate dehydrogenase component. That's the E3 domain. Here's arsenate. It's actually going to react with this. It'll actually form a bridge here. It'll lose two waters, and this will give us this complex right here. Now, this is called an arsenate chelate, and what's happening here is you have the metal chelate. Literally means chelate means claw, and so this would be an inorganic chemistry thing more typically, but you'll get a little bit of it here, very, very loosely. Think of it as a very, very, very stable metal complex, okay? And so you have this bonding to the arsenate right here. This right here would then be the 2,3-dimercaptopropanol. Is going to react with this. If we feed somebody in using to treat this arsenate, and typically this treatment is actually called chelation as well. Anyways, because you're, again, binding it with a new chelate. Anyways, this is the 2,3-dimercaptopropanol. You put that in, and what's going to happen is going to bond the arsenate. You're then going to excrete that because it's still quite soluble, and that will actually have restored the enzyme by removing those arsenate groups. You can actually use a very similar thing for same stuff really for mercury. You can actually, interestingly enough, use like glycol and stuff to actually deal with mercury, and the anything that has the mercapto, the hydrosulfide groups, actually bonds really well to things like mercury. So you can actually treat a mercury source with that. So the citric acid cycle, most likely assembled from pre-existing reaction pathways. Compounds such as pyruvate, alpha-ketoglutarate, and oxaloacetate were likely present early in the evolution for biosynthetic purposes. Which, if you thought about it, really makes a lot of sense because they're incorporated all over the biochemical pathways that we've already looked at, in fact. The thermodynamically favorable oxidative decarboxylation of the alpha keto acids is likely formed the core of the process that preceded the citric acid cycle evolutionarily. Glycoxylate cycle. This is a reaction sequence that converts acetyl-CoA from fat stores into glucose. This takes place in the organelles. This is called glyoxymes, prominent in oil-rich seeds like sunflower, cucumber, that sort of thing. This is similar to the citric acid cycle, but actually bypasses 2 carboxylation steps, and 2 molecules of acetyl-CoA enter the cycle to do this. And again, we're purposefully making the lipids from those carbohydrates. Remember that plants store their energy primarily in carbohydrates. because they have the room to do it. It doesn't matter that it takes up more space and it is much less energy dense. Don't care. They have plenty of room to do it. They don't have to run around. But if you have something like seeds that you want to attract things to eat, then you need to make them very sugary. If you want to reduce the size, you can then make more fatty acids, give them more energy for a smaller form factor. The sum of your reactions is 2-acetyl-CoA, NAD+, plus 2 waters, gives you succinate, 2 CoAs, NADH, and 2 H pluses. The succinate produced can actually be converted into carbohydrates by a combination of citrus acid cycle and gluconeogenesis. The glycoxylate cycle actually will allow plants, and it also shows up in some microorganisms, to grow on acetate. And so this is kind of a unique, interesting factor of this. Anyways, here's the cycle. You can see it's actually quite similar to the ones we've already done. It's just a little bit different. Even shows some of the same enzymes. Anyways, this is the isocitrate lyase, giving you succinate right here. And this is the glycoxylate right here, going in with a new acetyl-CoA. See how there's 2 points where you put in acetyl-CoA using 2 acetyl-CoAs for every cycle of this pathway. And then it goes back to the malate that you're more familiar with, malate dehydrogenase, giving you oxaloacetate, feeding you back into the more typical, what we'd already talked about, the citric acid cycle stuff. And so you can kind of see that. So some of the new treatments for things like tuberculosis, by the way, may actually be on the horizon. So tuberculosis, TB, is going to be a disease caused by Mycobacterium tuberculosis. This is actually one of the leading causes of death worldwide. It's transmitted by people with active lung infections by coughing and sneezing. It's commonly treated with antibiotic rifampicillin. Oh, man. One got me. Antibiotic resistant strains are emerging, which is why we continuously... You may have been wondering, like, okay, so honestly speaking, tuberculosis is one of those things. Is unless you're in an incredibly unstable region, it's not actually about wealth. People always get that wrong. That they say poor region, but it's not. It's not about that. It's about a region that nobody wants to ship to because they're going to have their truck stolen and lose everything and possibly killed. When you have very unstable regions like that, where either the government rule of law is not very good or the government themselves tends to seize things, you get a lack of ability to buy these things. No matter how much money you actually have, you can get them if you have enough money because people will smuggle them, essentially. Smuggling being avoiding anybody, even non-government entities, from seizing it. But anyways, tuberculosis is a huge killer in unstable regions in the world where you cannot get the medications. And you may be wondering, like, why the hell do we need a new treatment that may not actually be cheaper and may in fact be more expensive? The reason is that we're actually playing a game, a little bit of a chess match, if you will, against all microorganisms all the time. So the idealized microorganism is constantly trying to evolve to be able to infect us constantly and kill us very, 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 very slowly. It actually wants to leave us basically alive but suffering forever. So we can spread it forever. But those microorganisms constantly are evolving resistance to various different treatments. And so we constantly have to evolve and create new pathways of fighting them. For example, this being a new pathway that's a completely different one. There is a lot of antibiotic resistant strains that are emerging. This is a pretty bad one. And it's one of those that is definitely going to be more and more of a concern, and it's actually popped up in the United States in recent history. So M. tuberculosis is dependent on the gloxylate cycle, especially when they're latent stages in the lungs. By the way, if you're wondering where the social distancing and cloth masks and all that came from, it actually came from the treatment of tuberculosis and scarlet fever. It had nothing at all to do with virus transmission through the air, and was not at all useful in any way whatsoever against COVID. It was, even Fauci originally had said that, but he didn't want to panic people, and then he got political. So 2-vinyl isocitrate, this is a suicide inhibitor or mechanism that's based on the inhibitor for isocitrate lyase, and that's right there. When isocitrate lyase reacts with the inhibitor, succinate is going to be released, but thioester-linked homopyruvyl moiety actually remains covalently linked to the cis-192, inhibiting the enzyme. And so cis-192 is actually conserved in all strains of M. tuberculosis. And so this may actually kill off that entire enzyme's ability to work, and maybe a very, very stable treatment until basically tuberculosis would have to evolve enough to completely replace that enzyme and its functionality in that pathway, which may actually be more complicated than it sounds. It may actually have to do like 2 or 3 steps to replace that step, in which case it'll be a very long time before any sort of resistance develops against this, and may never. Tuberculosis may go extinct if these medicines can become cheap enough and widespread enough. So this is really a big deal, which is why it showed up in my lecture here. Anyways, you all have a wonderful time. I guess I'm going to call that a chapter, and we'll probably just start on a new chapter next time. Hopefully you get this. I guess it won't matter if you get this before, but hopefully you get it before. It'd make more sense. Okay, you have a wonderful day, a wonderful 2 days.